morning. How's everybody doing today? Hi, Sandra. Hi, Melody, Rasha, Trevor, Sarah, Anna. Good to see you. Hi, Imani. Good to see you as well. How's everybody doing this morning? Is my sound okay? Can everybody hear me? Looks like my sound is all right. Good deal. It's good to see everybody on this Thursday morning. Hi, William. Good to see you. Thanks for stopping in. I am absolutely wonderful. Better than I should be. How about that? One of my favorite TV personalities says that, better than I deserve. Okay, fantastic. So, this week we're talking about sensation and perception, and I didn't quite finish up Tuesday's lecture, so I'm going to start by talking a little bit about visual illusions, or not visual illusions, but organizing principles for vision. Okay, so let's go to the PowerPoint presentation. Let's see. So if you remember, we talked about the visual pathway. I suggested to you that information comes in through the eyes. It's perceived by these rods and cones, and then this photon energy is transduced into patterns of electrical activity. They travel down the optic nerve to the optic chiasm, where the left and right sides are transmitted to the correct sides of the brain, the correct hemispheres. Then goes through the thalamus, where it connects to your other senses, right? And then it comes all the way back to the visual cortex. Now, in the primary visual cortex, actually, the primary visual cortex processes the primary aspects of visual stimuli. That is, where's the object located, which direction is the object moving, what shape is the object, what color is the object. So that's where the primary characteristics of a visual stimulus are processed. Now, the thing about vision is it's not only sensation, sensing these qualities, but it's perception, putting them together and creating meaning out of these structures. And so, when we talk about our sensory perception, our senses, we have to talk about sensation as well as perception. Now, what your book talks about are the organizing principles of vision. And here's the crazy thing. You're born with these organs and with this area of the brain that has this ability to perceive color, shape, line, and all that stuff. But then you have to learn how to organize your visual world. And that's what happens during the first couple of years of life as you interact with the world and see that it follows certain patterns. So, if you will, perceptive processes are just abilities that we learn based upon repeated interactions with the sensory stimulus. So, when you hear sounds, at first you don't know what they are, but after a while you learn to differentiate a car horn from a boat horn from a train, and you learn to organize and name these things. Same thing happens with vision. We start out and the world is just a blooming, buzzing ball of confusion. And then we notice that there are patterns by which the world is organized, and we begin to use these patterns as guesstimates. So, in a sense, what we do is we go in and we put in our learned expectations over these sets of stimuli, and we begin to develop habits of perceiving the world. Now, in your book in chapter 5, they talk about the Gestalt organizing principles of vision. All right, these German psychologists in the 1920s began to understand that we had to think about sensory experiences in terms of their whole parts, their gestalts, rather than as atomic parts that we put together, right? So, um, if you would think, so we sort of divide the world in different ways. The first is that we divide the world into what we would call figures and grounds. The first thing you learn as an infant is to separate objects from backgrounds, figures from grounds. And that's sort of one of the organizing principles that human beings engage in. We separate our visual stimuli into things that are objects and things that we think are backgrounds or settings. But we don't just stop there. We actually have other learned habits of interpreting visual information. And your book's going to talk about five, 1, 2, 3, 4, 5 principles of grouping that humans use in order to organize objects in their visual world. Hello, Amin. Good to see you. Thanks for stopping in today. And so, your book talks about proximity, similarity, continuity, closure, and illusory contours. So, with proximity, we tend to think of things as being in groups of objects that are located close to one another. So, if you look at this picture right here, I have 5, 6, that's 11. I have 16 blue dots on the far left screen. What we typically do, instead of saying, look at that, there are 16 blue dots right there, what we typically do is we say, hey, look, there are 3 groups of dots located right here, a group of 6, a group of 5, and another group of 5. So one of the things that we tend to do is to group objects based upon how close they are. So you don't see 16 objects, you see 3 groupings of objects. This is a learned visual habit that we use to interpret our world. We also learn to interpret the world in terms of similarity. So we don't just see a whole blooming, buzzing confusion. Instead, we separate our visual picture into things that are similar. So if you look at image B, you'll notice that we have lines all over this page. Some of these lines are red, some of these lines are black. But if you'll notice, your attention is drawn to the fact that you have these squiggly lines that in my mind look like the seashore. And then you have these red lines that look like they're trying to depict water that's coming into that seashore. So if you'll notice, you don't just see a bunch of lines in B. Instead, you see 2 objects, squiggly lines and straight lines. So we group the world for visual perception processes into similar groups. So we group them into groups that have proximity and groups that have similarity. Now, if you look at the picture in C, now they've made the cylinder translucent to sort of give you the idea of what people do. But if that can wasn't translucent and you saw the green bar going behind it, what we wouldn't do is, if in actuality what you see is a pink uh, can, if you will, and then you see a green splotch on the left side and a green splotch, a green square on the right side. But what we do as humans is we make the connection between the left half and the right half. And instead of saying that we have a pink cylinder flanked by 2 green rectangles, what we tend to do is we say, hey, there's a pink cylinder and there's a long green rectangle that runs behind it. So we sort of uh, make things continuous even when they're not, right? So if you look at my arm, you don't see fingers over here and an elbow over here. You have learned that this is one structure that happens to be running behind my ears. Seems kind of stupid, but this is a learned way of interpreting the world. Good morning, dear Erica. Now, with D and E, these are demonstrations of where we create shapes out of outlines, out of things that aren't really there. So if you look at D, closure, now what we all see is a triangle. We all see an isolateral. Isolateral, is that right for my geometry students? We see an isolateral triangle here. Now, in actuality, the only thing that exists are 3 purple V's, if you will, that are oriented in a certain direction. But because we've seen triangles lots of times, what we do is we overlay the idea of a triangle on this image. So we close those lines and see a triangle, an equilateral. Thank you very much, Melody and William. I was a Bachelor of Arts and not a Bachelor of Science, so I didn't take that extra math class, right? And then you'll notice right below me, we have illusory contours. Instead of seeing 3 blue circles with pies, with pieces of the pie cut out, we actually see a square inside of this image. So we have added an image here. And so in a sense, A, B, C, D, and E, all of these organizing principles of vision our habits of interpreting our visual world. We interpret our visual world because it tends to organize itself in this way. And so when we go out and look for objects in our visual environment, we are trying to overlay this organizational structure. So this is where perception comes in. Sensation is having your brain stimulated. Perception is you figuring out what that stimulation means. And so these organizing principles are ways in which we organize and understand what it is we are looking at. Now, the same thing happens with depth perception. Now, if you see over on the far left side of the screen, I've got a phrase, binocular disparity, and a picture of two eyes. Here's the crazy thing. Your brain has a mathematical way to interpret distance for you. So if you look at these two eyes, they're sort of looking at two objects, A and B, and your left eye gets an image of the world that's a little bit different than the right eye. If you want to prove that to yourself, look at something close to you and then switch your eyes left and right, left and right, left and right. And what you're going to see is that object bounces to the left and the right and looks a little bit different. So your left eye sees one sort of shape of an image, and your right eye sees the other shape of the image. Based upon how different those two images look, your brain estimates distance. So true depth perception comes from what we call binocular disparity. Your two eyes see slightly different images. Those images are projected onto your brain, and your brain learns how to estimate differences to let you know how far things are away from you. Now, true depth perception is not possible when you only have one eye. And when I had my seated class, what I used to do is bring students in and I would put an eye patch on them, and then I would take ping pong balls and throw them at my students and have them try to catch these objects. And it was always really funny because my students have trouble catching these balls because they don't have true depth perception. Now, here's the deal. If you've ever gone to an art museum, and actually a lot of you probably know how to do this on your own, you can actually draw things and give the illusion of depth perception if you want. And so what's also been discovered is that there are what we call monocular depth cues. And again, these are learned because of the way the world typically looks. And we learn how to organize our field of vision and guess how far things are from you. So let's go to my whiteboard here. So here's the deal. Taking a look at this. Okay, taking a look at this picture. Let's see how straight this is. There we go. Now, I've drawn two mountains sitting on, and there's the horizon. Now, this is mountain A and this is mountain B. Can somebody tell me which one is closer, mountain A or mountain B? Which one is closer to us, mountain A or mountain B? I'll give you just a second. Okay, which mountain? Mountain A. And why do you say that, Amy? Tell me why you think mountain A is in front of mountain B. Why? Because you can see all of mountain A, but you can't see all of mountain B, okay? Great, fantastic. So what you're looking at is what's known as occlusion. Occlusion is one way in which we learn how to judge distance. Yes, the lines are overlapping B. Very good, Amy. Now, here's the other thing. I'm going to go ahead and put two people on this picture. There's Dave. And there's Fred. Which one of these people, so this is Fred and this is Dave, which person is closer to you, Fred or Dave? Fred or Dave? Fred or Dave? And not only tell me which one's closer, tell me why. Why is, you're funny, Dave. You're funny, William. Why is Fred closer? And you are all correct. Why is Fred closer? Two reasons. Two reasons. Melody's right. He is bigger. He is bigger. So we learn that people are about the same size. But when you see an object that's farther away, it creates a smaller image on your in your visual field. And we learn after a while that objects of the similar size, if one of them's much smaller, that must mean it's farther away. So the relative size of Dave, you know that Dave can't be a third of the size of Fred. Is there such a, I mean, there aren't that many 2-foot people running around in the world. Correct. He is closer, Trevor. We're asking, why is he closer? There's a second reason, too. The other is what we call height in the visual field. If you look at these objects, you'll notice that Dave is much higher than Fred. Now, given that this is a flat landscape, we know our brain tends to interpret things as being farther away, tend to be higher in our visual plane than things that are smaller. And so the two visual cues that your book's going to talk about are height in the visual field and what we call familiar size or relative size, if you will. Okay, so relative size of the one another. Now you can also use familiar size too. We know that Dave is supposed to be a certain size. There's no way he can be a third as tall as Fred, and as a general rule. And so we learn that sort of that, if you will, that visual cue. Now there's a couple of more. All right, I'm going to draw a road here and take a look at my road. Do you see my road? Boom, right here. Now, do you notice that as the road gets farther away from me, these two lines actually move together? Now, we all have learned that roads consist of parallel lines. These lines are supposed to be parallel, but the fact that they meet off in the distance allows you to guess the illusion of distance. So Dave is farther back here on the road here, and Fred is on the road here. And you'll notice that these parallel lines are closer together than these parallel lines. So we can tell that this road must be going away from us. And this is how we get the illusion of depth. And this is called linear perspective. And there's one more I'm going to show you. Now, you folks know when you drive around on a road, you see these big white hash marks in the middle of the road, differentiating the right line, the right lane from the left lane, right? Now, what you're going to notice is if you go out and look at a road that's going off into the distance, you're going to notice these white lines that are equally spaced out will start to become closer together and smaller. The texture gradient will change. And so that also, we know, we've learned that these lines should be the same distance away, but as we go off into the distance, we notice they look closer together. And so we begin to assume that there must be distance, that these lines must be farther away than these lines. So if you look at my wonderful art picture right here, you'll notice that I've created a picture on a flat piece of paper that has the illusion of depth. I put one mountain in front of the other. I've got one guy that is bigger, way bigger than the other guy, and he's also lower in your visual plane. I've got these parallel lines that seem to go together, and I've got this pattern that seems to get closer together as we move farther apart. Convergence, that's another way to put it. That's a great way to put it. And so, in a sense, what you're doing, what your brain is doing, these molecular depth cues and these gestalt cues are learned cues that you use to interpret what it is you are seeing in the world, right? So our brain's being stimulated the same way, but then there's this second process we call perception. And that's where you try to guess what it is you are seeing, hearing, smelling, or touching. And this process is learned, and this is sort of the place where your brain can be fooled by visual illusions. Now, not all visual illusions are perceptive illusions, but a lot of them are, and I'm going to show you some of those tonight in lab. But what I want you to take away from these last two slides is the difference between what we call sensation and what we call perception. All right, so you have a primary sensory cortex for vision, and that responds to color, line, direction, and movement. But then those sensory experiences are then moved to your association cortices that then try to guess based upon your prior experience what it is you are seeing. Does that make sense? Does everybody understand what I'm talking about? All right, that was yesterday's lecture that I didn't get around to finishing. So I'm going to go ahead and delete my slides real quick if you don't mind. And then I'm going to add some more slides for today's lecture. Okay, any questions? I know photographers, yes, absolutely, Melody. That's not good. If any of you ever watched, I read the Lord of the Rings when I was a kid, and was a big Lord of the Rings fan. And I know when they made the movie, in order to make Gandalf seem really, really tall, and these normal people to turn them into to hobbits and elves, they used camera angles, Melody, to make Gandalf seem much bigger than Frodo and Bilbo Baggins. You're absolutely right. I don't know if any of you have ever seen that 3D chalk art, the street art that 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 uh, that people do sometimes, but they can create these amazing images of depth and shape and size using camera angles. Absolutely. Ah, Trevor said your wedding photographer did that since your wife is taller. I hear you, brother. I'm 5'6". I remember in high school I was dating this girl who was 5'8", 5'9", and she wanted to wear heels to the prom, and I told her you absolutely cannot do that, and she said I absolutely can do it. So what I did was I sent away for a magazine on platform shoes, and I threatened to wear my own 3-inch platform shoes and look horrible at senior prom. And she relented and wore slippers, so I wouldn't wear my big platform shoes. Oh, you're 5'6" too. See, Anna, that's cool. But it's tough for us guys when we're 5'5", 5'6", man, because there's a lot of tall ladies around there, and it just looks strange if you're shorter than your girlfriend, right? You're 5'8". Nice. Your wife is 6'1". Oh, wow. Wow. I am A, jealous of you for being 2 inches taller than me, Trevor, and I am absolutely shocked and amazed uh, that your wife is 5 inches taller than you. That is crazy. Wow, Anna, your boyfriend's 6'1" and you're 5'6". That's amazing. That's a big size difference, and it's cool when the female's shorter than the guy, a lot shorter, but it's a little bit more awkward for us guys when we're not quite as tall as our female romantic partners, right? Depending on the angle, like the picture when you are putting your finger on top of the pyramids. Absolutely. Jesus, Noreen is 5'3" and her boyfriend's 6. The average guy tends to be 5'10". I've heard it's like 5'9". It might be 5'10", but don't make me feel any worse, Anna, uh, by saying 5'10". Okay? And females are 5'4". Right, right. This body dimorphism. Uh, Typically, the male of a species is going to be bigger than the female of a species. They call that body dimorphism. If anybody wants a 25-cent word to stick in their pocket today. It's only awkward. There you go. William, spoken like a true positive psychologist. It's only awkward if you make it awkward. Usually my ego put me at about 6'2", so I would stomp into a room at 5'6", feeling about 6'2". You know what I mean? I let my big old ego uh, carry me a few extra inches, but it still can be awkward sometimes. Okay, so let's talk about the other senses: hearing, taste, smell, and touch. We're going to talk about those for a few minutes today, and hopefully I can get all this in. Now, if you remember, this is what we talked about Confidence is definitely key, and I definitely push that chest way out, right? So if you remember, this is a slide I had from Tuesday. Remember, we have these primary sensory cortexes. Uh, the olfactory and the gustatory cortex are both located in your frontal lobe, so taste and smell in your frontal lobe. Remember, in your parietal lobe, you have your touch cortex, your somatosensory cortex. Remember, your auditory cortex is right here above your ear in the temporal lobe. And remember, crazy enough, your eyes are in the front of your head, but your visual cortex is in the back of your head in the occipital lobe. And if you'll remember, I suggested to you that not only do we have primary sensory cortices, okay, so your primary auditory cortices tells you pitch, tells you speed, tells you volume, but then the association cortex puts those sensory experiences together, and you learn to make guesses about what it is you are experiencing. So, for example, if you see me, I'm red. If you hold me, I'm smooth and I'm round. If you smell me and I smell slightly sweet. And if you take a bite of me and it's crunchy and tastes juicy and sweet, what is it that you are eating, folks? What is it that you are eating? It's red, round and smooth, slightly sweet smelling, and is very crunchy when you bite it. What exactly are you eating? And I'm going to let somebody guess. Dun, da, da. Boom. There you go, Imani with an apple. Absolutely, folks. It's an apple. Now, here's the deal. You only had those sensory experiences from your primary cortices, but then you had to put all those sensory experiences together and judge what it was you are actually experiencing. That's the difference between sensation and perception. You know, I have to admit, Eli, it could be a cherry as well. It could be a cherry as well. I'm an Apple fan, so it never even occurred to me. But yes, you are right, Eli. It could be a cherry. So on my next semester, I'll add maybe another sentence and say it's about as big as a baseball. Maybe that's what I need to do, Eli, but you're quite right. There we go. Trinity says apples. Or is that a cherry? See, I can't even tell. Pink lady apples rock. Oh my God, Anna. They are the most amazing thing in the world. In fact, I can't even really eat a Macintosh apple. You know, those kind of, because they're kind of soft and mushy in the middle. Ugh. Pink lady apples. I can even eat a Granny Smith, although they're a tad bitter for me, right? So let's talk a little bit about hearing for a second. If you look in your book, you're going to notice that you have this little structure. Uh-oh, let me make my picture smaller here for a minute so that I'm not in the way. Okay. I got my inset picture here. There we go. There we go. Okay. So, um, oh, cool. I'm glad you are here, Gloria. Thanks for coming. Gala apples are pretty good too. I definitely have to give you credit for that. Gala apples are pretty dadgum tasty. And let me move my image is all funky over here. Okay, so let's talk a little bit about hearing. Now, you're going to see in your textbook for each of your other senses, actually for your visual sense too, you're going to have these little inset pictures. And this is kind of what I want you to focus on for the homework and the test, because these little pictures sort of explain the basic process of these sensory organs. So you have peripheral organs, and then they transfer to the central these organs that are located in your periphery, part of your peripheral nervous system, and they transmit the information via a nerve to your to your central nervous system and a primary sensory cortex within that nervous system. So let's talk about the ear first. You'll notice we have a picture of an outer ear here. The big pinna is responsible for capturing sound and sort of moving it into the ear canal, where it travels down the ear canal and makes this eardrum vibrate. You have this thing called an eardrum in your outer ear. We've all probably heard of that. You may have even had a friend who had a burst eardrum. The idea behind the eardrum is that these sound waves when they come in, they make that eardrum vibrate. You then have three teeny tiny bones in your middle ear. Can anybody tell me? Oh, never mind. The answer's already here. You've got these three tiny bones in your ear. These are the smallest bones in your body. The incus, the stapes, and the malleus, or more, which are their Latin names, or you can think of them as the hammer, the anvil, and the stirrup. And you have these three little bones, and these bones vibrate. They're attached to your eardrum, and when your eardrum vibrates, these little bones vibrate. And these bones then connect with this structure you have called a cochlea. And on your cochlea, you have a little bitty hole called an oval window. And what happens is this oval window and these three little bones, they magnify the vibrations that your eardrum feels by, I think it's a factor of 10, I think it. So the vibration that is created on your eardrum from sound waves is magnified by these three bones that then touch this thing called an oval window located on your cochlea. Now, the picture in B is your cochlea. Now, for demonstration purposes, they've pulled out your cochlea and they've sort of unwrapped it, if you will. Unwrapped it, if you will. Right? But it's basically a little semicircular canal filled with fluid and hairs. There's this thing called a basilar membrane that runs down. It's wrapped. You see this, and you have this basilar membrane that runs down the length of the cochlea. And on this basilar membrane, you have these hair cells. Now, the cochlea is filled with fluid, and it have this membrane with these hair cells on it. And what happens is when your hammer, anvil, and stirrup vibrate on that oval window, it causes the fluid in the cochlea to move. And the fluid causes these hair cells to bend. And that's what turns sound waves into patterns of electrical activity. We'll call that process transduction. Remember, we talked about transduction. So, sound waves are transduced into patterns of electrical activity in the cochlea because the sound waves cause these hairs to vibrate. And the vibration of these hairs then is transmitted to your auditory nerve, you see, and your auditory nerve goes through your thalamus. Remember, all senses go through the thalamus except for smell, and then it travels to your primary auditory cortex. Notice that you have a left primary auditory cortex that hears what your right ear is hearing, right? And your right primary cortex is hearing what your left ear, the sound that your left ear is hearing. Okay, so you have these hair cells, and then you have these semicircular canals, which is sort of the inside of this cochlea structure. Hearing loss is due to hair cells getting damaged. Yeah, the weird thing is, I actually experienced some hearing damage. I've got a ringing in my left ear. I had to take chemotherapy for the cancer last year, and it turns out that chemotherapy, one of the side effects that occurs to some people is hearing loss. So I've got this ringing in my ear and some hearing loss, and they're not exactly sure why one hears the ringing in their ears and why this causes you to lose your hearing ability. Now, you can lose hearing ability because you damage the eardrum, and you can have hearing damage because you damage these hair cells in your cochlea. But nobody really 100% understands hearing loss. That's a great point, Gloria. Hearing loss can occur for several reasons, but it does relate to those hair cells and their vibrating ability. Tinnitus. Tinnitus gives me that high pitch ringing all the time. I know, right? I hear it. When things get quiet, I hear my ears ringing, and it can be quite annoying, Trevor, right? Now, so the cochlea is a fluid-filled, rolled-up tube containing the basilar membrane and the hair cells. This is where the qualities of sounds are transduced. Now, really, in a sense, your brain hears two basic qualities. It hears pitch, and it hears actually three qualities: pitch, loudness, and sound localization, where sounds come from. Okay, now let's talk about loudness and localization quickly, and then I'll get into perceiving pitch. But loudness is generally determined by the sound wave amplitude. So the bigger that sound wave, so when we talk about waveforms, you look at this waveform right over here on the picture, okay? The height of the wave going from crest to trough, okay, is what determines how loud that sound is. And louder sounds create more pressure on the eardrum. More air pressure is perceived by you as loudness. It's generally determined by the amplitude of the sound wave. Now, here's a crazy thing that we learn. This is a perceptive process. It turns out that our ears are located on different sides of our head. So when you hear a sound, the ear that it's closer to will hear a louder sound, and it will hear the sound a little bit earlier than the other ear will. And so your brain takes these two qualities, the difference in loudness in one ear and the other, and the timing at when sounds reach your ears, and learns after a while to judge where sounds are based upon these two qualities. So I'm snapping my fingers over here to the right. Okay, my right ear hears the snapping louder than my left ear, and my right ear hears the snapping sound just a nanosecond before my left ear does. And over the course of my lifetime, I have learned how to interpret that and figure out where objects are located in my environment. Okay? Now, how do we perceive pitch? And pitch is whether or not something's a high C or a low E. That's the best I can sing, so don't ask me to do any more. High, low. How do we differentiate sound pitch? Well, your perceiving pitch in your ears is determined by how close, how small the wavelengths are. So wavelength, if any of you have ever sat in a physics class, wavelength is the distance from the peak of a wave to the next peak of the wave, or from the trough, the bottom of the wave, to the next trough. So the closer the peaks are together, the higher the pitch sound is. The farther the peaks are away, the lower the pitch is. How do we transduce this wavelength into pitch? All right, now there are two basic theories. Really, it turns out that you can think of place coding as probably the more accurate theory of how sound is transduced. Now, some people suggest that the faster the hair cells vibrate, okay, the higher the pitch is, and that's what we would call temporal coding. So the faster the hair cells vibrate, the higher the pitch. Now, really, the evidence actually leans more towards what we would call place coding. And what this tells us is it's not so much how fast the hair cells vibrate, but which hair cells vibrate. So if you go back to this picture, you're going to notice that the semicircular canal located in excuse me, not the semicircular canal, but the cochlea. The cochlea is this long structure, and it's got hair cells at different places in it. And so what your book suggests is that where these cells vibrate determines the pitch that you hear. So when you hear high-pitched sounds, it makes the hair cells at the very beginning of the cochlea vibrate, and this is perceived by your brain as a high-pitched sound. And what they suggest is that low-pitched sounds cause the hair cells at the end of the cochlea to vibrate, and those are perceived as low-pitch sounds. So that's how sound is perceived. Now, really, if you think about it, we perceive pitch, loudness, and location of sounds. Those are the things that we sense. We then have to learn how to interpret the sounds we hear, and that occurs in association cortices. So your auditory cortex connects, just like other sensory cortex connects with your with your memory structure. So you hear that little tweet sound, and you, wow, I remember that sound. I saw it one time when I saw a bird. So that sound must be a bird tweeting. And so there's this perceptive process, and there's also this sensory process. Now, let me go back, and I mentioned this earlier, but I said it a little sloppily. You then, it turns out that you also perceive, you have a sense of balance. Not only do we hear, see, smell, taste, and touch, but we also have a sense of balance. That is, where is my work body in space? How am I oriented? I am not upside down right now. I am right side up. How do I know that? My semicircular canals are telling me that. I am now sideways. How do I know that? My semicircular canals are telling me I'm sideways. And some people who have damage to the semicircular canals will have balance problems and may even feel nauseous because their semicircular canals aren't working right. Now, the semicircular canals, just like the cochlea, they're filled with fluid. However, you have three semicircular canals and they are all oriented in different directions. And each of these semicircular canals has fluid, and they also have hair fibers. And so as your head turns left and right, up and back, this fluid runs around these semicircular canals, bending these hair cells. And the pattern of bending is interpreted by your body as head position. So when I turn to the side, these fluid in my semicircular canals runs in different directions, bends these hair cells, and that information is interpreted by my cerebellum. So here's the deal: my hearing going out of my basilar membrane located in my cochlea, okay, goes to my auditory cortex, and that gives me sound. But my semicircular canals connect via a nerve pathway to my cerebellum. And if you remember earlier in chapter 2, I suggested to you that your cerebellum was important for balancing and smooth motor movements. So balance comes from your semicircular canal feedback, the fluid floating in and bending those hairs in your semicircular canal, and that is then interpreted by your cerebellum as balance information to let you know which way your head is facing. There you go, Trevor Braverman with the vertigo. Absolutely. Is vertigo determined by your semicircular canals or problems with your cerebellum? I'm not 100% sure, Trevor. So if you know, feel free to type in the chat bar and give us the explanation for vertigo. If you don't, I'm going to be obligated after class to look it up myself so I know it, right? Okay, so that's how hearing. Oh, and I do want you to know they're called mechanoreceptors. Remember, you have visual receptors in your eyes. Your ears have what we call mechanoreceptors. That is, they turn pressure information into sensory experiences. Why it doesn't seem to bother them? You know, that's a great question. And it's interesting you should mention that. And if somebody reminds me tonight in webinar, I will show you this. Babies hear high-pitched sounds better than older people do. In fact, as you get older, your ability to perceive high-pitched sounds actually goes away. I don't know if any of you know this, but a while back, somebody invented a ringtone that could only be heard by younger people. And so kids could sit in class and have their cell phones ring with messages, and the old teacher in the front couldn't hear it, right? Um, and so, in a sense, as you get older, your ears lose the ability to hear high pitched sounds. And the crazy thing is, I could actually tell you your age by the sounds you cannot hear. So tonight, if you come to webinar, I'll play that little audio clip for you. And what it does is it has a pitch starting low and going up high, high, high. And you will find that at a certain point, you will quit hearing the sound. And that's based upon how old you are. So infants are more their brains hear higher pitched sounds, Imani, and so maybe that's why they like them. On the other hand, high pitched sounds drive us old people crazy until they get so high we can't hear them. Have you folks ever heard of dog whistles? It's those whistles that blow at such a high pitch that humans can't hear them. Same thing, except as you move through your life, you become less and less able to hear these high pitched ends of the sound. You start losing that ability. Hearing declines as we age, absolutely. And lack of hearing is also going to be related to social disengagement. So people, as they can't hear, they start, and when we talk about, in my developmental psych class, I talk about the effect that the senses have on our feelings as we get older and in our behavior. And what you're going to notice is that as people's hearing declines, they tend to socially disengage from the world. And that's a great point to bring out, Gloria. And vertigo has something to do with the calcium deposits in your inner ear moving. Ooh, thank you, Amy. All right. So Trevor was a fight in the Marines. He got hit in the ear and was told that's what caused my vertigo. All right. So it sounds like we're building up a theory of hearing loss and of balance problems here in class. Thank you, Amy. Thank you, Gloria. And thank you, Trevor. Right? Is it the same for deaf people? Right. Um, that's a great question, and it depends on whether or not you're born deaf or whether or not you experience, you have an event that causes you to become deaf later in life. So we typically, when we talk about sensory loss, we tend to talk about peripheral damage versus central damage. And it would probably be people like myself who listened to too much rock and roll and then had chemotherapy. My hearing loss is peripheral. It's related to what's happening in my ear. On the other hand, some people may be born with a problem with the nerve or the area in their brain that responds to it. And so that might be central nerve problems, central nervous system problems. So it can happen because of either reason, Gloria, and it depends on why you were deaf and when you became deaf. Another great question. Another great question. All right, let's talk about taste and smell. Taste and smell are carried by chemoreceptors, and a chemoreceptor's job is to transduce chemical information into patterns of neural activity. So remember we talked about transduction, and your eyes transduce photons into patterns of neural activity. Your ears transduce sound waves into patterns of neural activity. Chemoreceptors in your mouth and in your nose transduce chemicals into patterns of neural activity. Now, in your tongue, on your tongue, you have these taste receptors, and these are receptor cells that transduce chemical stimulation into action potentials. Taste receptors are not taste buds, but taste receptors are located in taste buds. Now you have 8 to 10,000 taste buds in your mouth. These taste buds have these chemical receptors, and these chemical receptors, you have hundreds of different types of chemical receptors that respond to different tastes. And this travels along your facial nerve. See if you'll notice in the picture right below me, it goes from the bottom of your tongue down through the bottom here, and it goes up into your gustatory cortex in your brain. It goes through your thalamus. Now, here's the deal. There are some people who don't like broccoli or coffee because it's too strong. Are you one of those people that hates broccoli or coffee or strong tasting stuff? If you are, you might be a super taster, a super taster. And if you look in your book, there's a demo to help you determine whether or not you are a super tasters. And super tasters have 6 times as many taste buds as normal people. And so these folks are often more sensitive to tastes. And these are going to be some of the people that don't like these stronger tasting foods. I love collard greens. I love sour pickles. I love coffee. So I am clearly not a super taster. But if you're one of these people that doesn't like really strong foods, you may be a super taster. Oh, Noreen doesn't like broccoli. Holy cow, we eat broccoli about 4 times a week in this household. It's a superfood, man. So I eat broccoli. In fact, I had broccoli for dinner last night. It's a good thing you don't live over here, Noreen. You'd probably starve, right? Now, here's the deal. I take that back. I was talking about taste, smell when I said hundreds of different smells. With taste, you have 5 main tastes. So you have a sweet taste, a sour taste, a salty taste, a bitter taste, and an umami taste. And these tastes are sort of perceived in different parts of your tongue. So you have sweet tasters. So some of your receptor cells respond to sugar molecules, glucose, sucrose, fructose, any of these oses. You have certain taste receptors that respond to these sugar molecules. And this gives you the sense of sweet taste. And you're going to notice that your sweet taste is right here on the tip of your tongue, along with salty. Salty is also on the tip of your tongue, and your taste receptors that respond to sodium ions give you the sense of saltiness. So whenever you taste sodium chloride, you will get the experience of saltiness. Tears have a little bit of sodium in them when you hear it. When you cry a lot and the tears get in your mouth, you taste salty tears. That's because your taste receptors are responding to sodium ions. Okay? Coffee is life for me, Trevor. Spoken like a true Marine. There you go. They fight and they drink coffee. That's what them dadgum Marines do. Now, hydrogen ions, your taste receptors that respond to hydrogen ions give you a a sour flavor, a sour flavor. So if you've eaten pickles or anything that's sour, your tongue, your taste receptors are responding to hydrogen ions. Now, there are several different chemicals that your brain responds to for the taste of bitter. And you'll notice bitter is at the back of your mouth. And basically, lots of things that happen to be poisonous also taste very, very bitter. So some people believe that the taste of bitter was created, evolved as a protection for you to avoid eating poisonous things. A lot of the things that are AKA poisonous to you also have molecules that create the sense of bitter in your mouth. So some people, and you'll notice bitter is right there at the back of your mouth where you can spit it out before you swallow it. So some people think that the taste of bitter evolved to sort of keep you safe and prevent you from eating things that were poisonous. Now, you've probably all heard of sweet, sour, salty, and bitter, but I'm going to talk about a 5th taste called umami. And this is what happens when your sensory receptors that respond to glutamate are activated. If any of you have ever had pot roast, right? That real gravy kind of pot roast, or if you've gone to Thanksgiving dinner and you've got gravy and stuffing and this rich chicken and turkey that you're eating, those tastes, those meaty kind of tastes, are called umami, and your brain gets those when your glutamate sensory receptors are activated. And so these are the 5 basic tastes. And then, as I suggested, there's this perception process where your brain learns to interpret patterns of these different flavors together to know, hey, I'm eating watermelon. Hey, I'm eating orange. Hey, I'm eating turkey dinner or whatever. So you have these five basic tastes, and then your brain learns to interpret these later on. Now, I do want to point out another thing again, and I don't want to talk about this and beat it into the ground. I don't think it's bad, and I definitely don't want your pity. But part of the radiation therapy to my throat destroyed my sense of taste. And I talked to the doctor, and I said, "Well, is the radiation is it destroying my taste buds?" And the doctor was like, "Nope. What it's causing is swelling underneath your tongue." In this neural pathway. You see this neural pathway down here right below me that goes from your tongue all the way up to your brain. Well, what happened when they radiated my throat, it caused swelling down here, and this swelling damaged all the nerve fibers that were running from my tongue. My taste buds were still working, but the nerve fibers that carried taste to my brain were all damaged. And for about 3 months, nothing tasted like anything. I couldn't taste a single thing. It all tasted like paper to me. And then my senses started coming back, but not at the same time and not all together. And then things started tasting really bizarre and weird because I wasn't getting the right combinations of flavors that I thought. Now, my taste has almost come back after about 10 months, but the only thing I can't taste, folks, I have no sense of taste for sugar. When I eat a candy bar, I can't taste the sugar. Snickers bar tastes like they're made out of sand to me. When I eat ice cream, it tastes like sand to me. So I don't eat candy bars or ice cream anymore because I just can't taste them. Now, I talked to my doctor, and he said that if your taste doesn't come back for a year, you're probably not going to get it back. So it's been almost a year, so I would say that for the rest of my life, I will never ever be able to taste ice cream or chocolate. How many of you could, oh, great question. Gloria, would taste buds be less effective if you lost your sense of smell? No, they would not be, Gloria. No, they would not be. But the question you're asking is a perception question. When we determine what something tastes like, we interpret what our taste buds tell us, and we combine that information with what our nose is telling us. So it's not that your taste buds aren't as effective, uh, Gloria, but your taste buds are supported by your nose buds, if you will, by the receptor cells, the chemoreceptors in your nose. So if you were to pinch your nose closed and eat an apple, and take a bite of an apple in a potato, what you might find is that the apple and the potato, without a sense of smell, your taste buds are going to tell them, they're going to give you primarily the same sort of feedback. And you're going to have trouble telling the difference between an apple and a potato. So the difference comes when your nose. So your taste buds still work, but as I suggested, there's sensation and there's perception. The final perception, the knowing what it is you tasted, is dependent upon how it smells as well as how it tastes. That's a great question, Gloria. So, Noreen lost her sense of taste when you got COVID. It's obnoxious, isn't it? It's crazy. I didn't realize how important taste was to hunger. When I couldn't taste anything, I had trouble eating and I had trouble gaining weight. I actually lost 30 pounds during the chemotherapy. Part of it was from pain, but part of it was from everything tasted so nasty I couldn't eat. It was really, really crazy. Okay, fantastic. Okay, we are at 9:58. Dadgummit, I didn't get to smell and I didn't get to touch. We're not going to have time to cover those things. So if you would, I want you to do this week's homework by reading the smell and the touch slides on your own. Go to the book and read those inset pictures that I showed you that are down here that exist. And hopefully you can learn a little bit about the sense of smell and the sense of taste, okay? Oh, Noreen has an appointment to get to. We all have an appointment to get to. I am so sorry for keeping you 59 minutes. Folks, have a great day. I will see you tonight in class at 7 o'clock if you can make it. Otherwise, I look forward to seeing you next Tuesday when we talk about behaviorism. Hey, folks, have a great day. Bye.